Hi, this is Mrs. Bustamante. Today we're going to continue our notes on DNA replication. We're really going to walk step by step on the process and all of the molecules that are involved that you should know and be able to explain through that process. So remember when we're doing DNA replication, we're starting with a DNA molecule that is double-stranded and is actually in the form of a double helix. So it's twisted, but we're going to draw it as kind of a straight line because it's a pretty complex process and hard to understand. Here are all the different molecules. You'll notice lots of them end in ACE because they're enzymes that we're going to use in the process of DNA replication and that you should know and understand and be able to explain their purpose in the process. So the first thing we have to do in DNA replication is we have to cut apart this DNA because we need it to be able to replicate one side and the other. We basically want to end up with two copies of it. And the thing responsible for that is something called helicase. It's here in green. And I like to say that helicase cuts, because it has this C in it that helps me remember it, the double-stranded DNA into two single strands. And why can it do that? Well, remember these bases are held together by hydrogen bonds, and hydrogen bonds are not that strong. So this is actually a beneficial point when it comes to replicating the DNA because we can split those bonds pretty easily. We can cut them apart. But they do actually want to bond together, so we have to have something that helps them stay apart. And that's that purple molecule right here. It's called DNA binding protein. And its job is just that, to hold that DNA apart, to keep it as single strands, so that we can move forward in the replication process. The second step involves something called RNA primase, and it's going to add RNA complementary bases to the DNA strand. You'll notice in this picture, this top part provides us a good image. The DNA has AA, and you can see that RNA primase is adding UU. And that's a really good indicator that this is RNA primase and that this is RNA. Because remember, all the other bases are the same, except for RNA, uracil replaces thymine. And the reason we have to have RNA primase is DNA polymerase that will add our DNA nucleotides, complementary nucleotides, can't just go ahead and start off on the DNA strand. We have to add kind of this buffer first to it. So the next step is DNA polymerase. DNA polymerase is going to continue to build our complementary strands, adding on to the RNA bases, except it adds DNA bases. And this is nice because we can start to see it happens in both directions. So up here, right, in our picture before, we saw that we had RNA polymerase, and it added those bases to, RNA primase, it added those RNA bases to it. But we can see that both strands actually have some RNA, and then we have DNA polymerase that is adding DNA nucleotides, complementary ones, the ones that match, right, based on our base pair rules, to the original strands. Now you're going to notice that they go in opposite directions. And there's a reason for that, but we don't have to worry about that right now. The one that we see gets built continuously. It looks like a straight line. It looks like there's no interruptions in it. We call the leading strand. And it is really built in just one piece, minus these little RNA parts, right, that DNA polymerase needs to build off of. And then we have the lagging strand, and it's our strand down here below, and you'll notice that it kind of gets built in segments. So right here's RNA primase, and then we're going to have DNA polymerase that'll come in and add these, and we had a little section of RNA because DNA polymerase had already added those nucleotides there, and that gets built in segments. We call those segments or those fragments Okazaki fragments, just after the person who discovered them. But it's important to know that these two things are happening at the same time. Your leading strand and your lagging strand are getting built simultaneously. It's not one and then the other. They happen at the same time. So the DNA strand is getting built in both directions at the same time. You'll notice on the animation we walk through, there are these little dark circles. That's a DNA polymerase again. And it's going to go in and replace those segments of RNA. I'm not going to require that you understand that and know that for now, but just so you know, that's what's happening. The last step we need to know is these dark rectangles, and they're going to go in and they're going to link together these holes, right? Because there are some holes from where we had primase. We need to complete our sugar phosphate backbone, so they go in and they fill in those holes. So what do we end up with? When all this is done, you'll notice that we have two identical double helix strands of DNA. Half of the DNA came from the original strand, this very top strand right here, and this very bottom strand right here, and half of that DNA is newly synthesized or newly made, and that's the bottom strand here. The inside strands, I guess, maybe are a good way to say that. And we add complementary base pairs are added to complementary strands. So what that means is because we're just matching things, you will notice that these two strands of DNA are exactly identical. You cannot tell them apart. And that's because of the process of how they get made. We call it the semi-conservative model. That's how we describe how DNA is replicated or DNA is copied, if you will. So I want you to go ahead and watch and work through the animation and explain the animation to yourself, because what's hard to see in pictures like this is that this is a continuous process. It flows from one step to the next without stopping. It just goes and goes and goes. And the animation does a nice job of showing you that. If you want to look at some other animations, this is another great one to view, and I'll post that link as well.